is Roare Estrada, and this group will be discussing figure number 3, sections I through L, from the journal article entitled Human iPS Cell-Derived Dopaminergic Neurons Function in a Primate Parkinson's Disease Model by Tetsuhiro Kikuchi et al. These figures will be discussed by Miranda Garnica, Erin Melville, Minerva Rivera, Fidel Yanez, and myself. So the purpose of these figures was to explain results regarding the success of iPS cells actually functioning as working dopaminergic neurons, as well as explaining the correlation between the Ki values of the primate models with the treatment of iPS cells from various origins, such as humans with Parkinson's disease or healthy humans as well. The general workflow goes as such. The primate models were first injected with human iPS cells. They were then monitored over the course of 24 months. And over this course, the primate models were screened using positron emission tomography, otherwise known as PET scanning, for F-DOPA and DAT. Magnetic resonance imaging was also used to monitor the putamen region of the brain and monitor the size. The Ki scores were then calculated and graphed in order to analyze comparisons of the values, the numbers of the Th cells in the putamen, as well as the time elapsed.
Hi everyone, my name is Minerva and I'm going to be going over some terms that are commonly used in this letter research. That way we have a better understanding of what's going on. So first we're going to look at figure 3I. These are PET and MRI scans. Scientists use PET and MRI scans to determine whether grafted cells are capable of functioning as dopaminergic neurons. Using these scans, scientists examined dopamine synthesis of F-DOPA and found an increase of it in the putamen. So firstly, what are PET scans? PET scans, or positron emission tomography, use image testing to reveal how your tissues and organs are functioning. This is done by using a radioactive drug or tracer. MRI scans, or magnetic resonance imaging, form pictures of the anatomy and physiological processes of the body. This is done by using field gradients and radio waves to generate images of the organs in the body. So I also mentioned F-DOPA. F-DOPA is a fluorinated form of L-DOPA and is used as a radio tracer in PET scans. And fluorinated simply just means to introduce fluorine into a compound. So moving on. We're going to talk about L-DOPA. So L-DOPA is an enzyme that is naturally found in our bodies and is later converted into dopamine. Now moving forward, this is figure J, K, and L. So these figures each showcase Ki values in the y-axis, and this is obtained from F-DOPA using PET scanning. So in these figures, Ki values represent the amount of F-DOPA in the putamen. And the putamen regulates movements and is connected to the substantia nigra, and therefore also houses dopaminergic neurons in the brain. And lastly, Th positive cells, or tyrosine hydroxylase, is an enzyme that's responsible for catalyzing L-tyrosine into L-DOPA. So these cells are simply responsible for producing the enzyme tyrosine hydroxylase. Thanks.
Okay, hi guys, I'm Erin Melville. I'm also in this group, and I'm going to be going over the results and conclusions for figure 3I. So this is the first figure in figure 3 that our group has assigned, and instead of being a chart or a graph, it's actually the physical photo results of the MRI and PET scans from one monkey over time. So the first one is with the initial treatment of MPTP, which creates the symptoms of Parkinson's disease. And then the second row, or the second column, sorry, my bad, is after 1 week, and then 3 months, 6 months, 12 months, 18 months, and 21 months. So this is showing the progression over time of the certain area in their brain that is of interest to the researchers. So the first two black and white rows on top are MRI results. And the white dotted line is outlining the area of the brain called the putamen. And this area is what scientists are interested in studying for this experiment, because that's where the L-DOPA and the dopaminergic neurons, that's where they're concentrated. So that's the area of the brain that is really important to them. And so the more you can kind of see, especially in the second row right there, how over time there's more white inside that area. And so that means there's more functioning dopaminergic neurons and more things filling up the putamen, which is good. That's what they want. And then the third row and the fourth row down, the colorful ones, are results from PET scans. And these both screened for different things that both indicate the survival of the dopaminergic neurons in the monkey's brain. So the first row, or the third row actually, but the first row of PET scans is screening for F-DOPA, and that is a fluorescing version of L-DOPA, which suggests the survival of dopaminergic neurons because it's being produced, it's being put out there, and because they made it fluoresce, then we can pick it up on a PET scan and it looks like that. So you can see that the more time progresses, the more dopamine is being released and created, and that means that the dopaminergic neurons are surviving, which is really great. The CPE2I test also screens for something called DAT expression that is going to also indicate the survival of dopaminergic neurons. And I definitely know a little bit less about this than I do about F-DOPA, but I just know that it's kind of another, it's not like a control group, but it's just another backup, I guess, to make sure that the F-DOPA results aren't off. So since both of those are separate chemicals, DAT and F-DOPA, they're separate, then if we screen for both and they both come back that they're both suggesting the survival of dopaminergic neurons, that's twice the proof, and that's better for this experiment.
Okay, so I'm Miranda, and I had the results and conclusions of figure 3J and K. So as you can tell, J and K are both two graphs that whose variables is the Ki values of the monkeys and versus the time after transplantation. And they're two line graphs, but they also have a little bit more information on them other than that. So basically, they tested these K values and calculated these K values through the DOPA-PET test. And these Ki values significantly correlated with the number of surviving Th plus cells, which actually were grafted and suggest that the survival of the DOPA neurons can be monitored through this test. F-DOPA is simply an indicator that suggests the survival of the neurons. These two graphs both have a few similarities and differences. On the left and right side, they both have gray dotted lines, which basically that is the average Ki values for two intact monkeys. So they should be around there for full recovery. Also the variables, as I mentioned, are the same for both graphs. And the biggest differences are the time span and the categories of the graphs. On the left-hand side, they only input the data for 12 months versus the right-hand side, which goes up to 18 months. The categories of the monkeys as well. So on the left, you can see that there's the vehicle group, the healthy group, and the Parkinson's degree group. But on the right, you can see that all 10 monkeys are represented, or from the 3rd to the 10th, and they're not in order, but you can see that they're color coded, and this is the key for it. The results of these two graphs, they basically found a gradual increase of F-DOPA through these tests. The Ki values of the normal monkeys were 7.1 plus or minus 0.18. The MPTP treated monkeys were 0.84 plus or minus 0.6. And the transplantation monkeys were 3.5 plus or minus 1.9. And so what this shows is that the Ki values of the transplanted monkeys recovered to about 48% of the levels found in normal monkeys. And this just indicates how well the dopa neurons are working. And so you can see that 3.5 is about half of 7.1. So this indicates like good growth in their brains. And yeah, that's what they concluded from that.
Hey guys, my name is Fidel, and today I will be presenting to you figure 3L. First things first, this graph represents the correlation between the surviving TH cells and the Ki values from 18F dopa PET. And I know some of us may not know what 18F dopa PET is, so I took the liberty of doing some research, and it actually is the agent that measures the uptake of dopamine precursors for assessment of presynaptic dopaminergic integrity. And it has been shown to accurately reflect the monoaminergic disturbances in Parkinson's disease, which is really cool, and is why they're using it in this experiment to begin with. The influx constant, also known as the Ki values, for this graph was calculated using the pot-like reference tissue model to obtain quantitative values in the transplanted areas of the monkey's brains, which is also really cool. The values for the surviving TH cells for the healthy group of monkeys is in blue, and as you can see right off the bat, the surviving TH cell values were just really high in the 5s and in the 7s even, and that could be due to the monkeys being healthy already and being able to produce TH cells without having something helping them produce it. The red dots in the graph shows the values of the surviving TH cells in the Parkinson's disease group. And even though they start off really slow and really low, you can see that throughout the minutes in the experiment, they actually go up and end up surpassing the blue dots, which is really good because that's what we want from this experiment. We want to see if the number of TH cells on the sick group is able to survive and reproduce faster. All right, the linear regression line shows how two groups did significantly good at producing TH cells throughout the experiment because, as you can see, the line goes right through the middle of the two groups, and it's going up, meaning that both groups are doing really good with the TH cells, keeping them alive and just keeping them up. The Ki values significantly correlate with the number of surviving TH cells in the graphs, suggesting that the survival of dopaminergic neurons can be monitored by 18F dopa PET, which is really cool, and which is why we're using 18F dopa PET to begin with. And now we know more, and we'll keep going from here.
And for our last slide, our group wanted to go over some questions that we had regarding this letter research paper. The first being, why did transplanted monkeys only recover about 48% of the levels found in normal monkeys? As we look at figure 3L, we again see that transplanted monkeys only recovered about 48% in comparison to normal monkeys. And so we just wanted to know why that is. What factors are affecting transplanted monkeys? Is it because they went through surgery, or is it something else? Secondly, we asked, are there different methods scientists can use other than PET scans or MRI scans to determine if grafted cells function as dopaminergic neurons? This is because PET scans use radioactive rays, which can be harmful, and therefore PET scans can only be done a certain amount of times. And MRI scans also come with its own risks. Like, for example, if you want to sedate the patient, that sedation drug also comes, with its own risks. And lastly, we asked, are there plans to move forward into human testing with this experiment design, and if so, how? Thank you.
Thank you guys so much for watching our video and listening for this whole 15 minutes. And we hope you learned something from figure 3 I through L. So Benjamin says, see you next time.